Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. I'm very happy to welcome Solchan Won to the club today to talk to us about some recent work that was published in the Journal of Extracellular Vesicles just very recently. And so Solchan is joining us from Seoul National University in Korea, and has some very interesting work here on some EVs that are not coming from mammalian sources, but rather from bacteria, and how they can be used as well to fight cancers. So Solchan, thank you so much for joining us today, and we're looking forward to your presentation. So I'd just like to invite you to go ahead and get started whenever you're ready.
Okay, thank you very much for your kind introduction. So hi everyone, my name is Sol. I'm a PhD student in Seoul National University in Dr. Lee's lab in Korea. Um, it's an honor for me to have an opportunity to present our recent work in front of expertise of EV field, and I'd like to thank the organizers for having me in this presentation. Right, so I would like to begin with a brief introduction of what we do and what we normally concentrate on in Dr. Lee's lab. So these are our recent published papers. And as you can see in the title of each journals, we utilize a variety of substances from small molecules to viruses that are capable candidates of potent cancer immunotherapy. And we kind of focus on the mechanisms of immune responses within the tumor microenvironment. So before we dive in, I would like to share some key background information about important topics in cancer immunotherapy that we have focused on in this research. So this diagram demonstrates how T cells get activated and are differentiated into memory or effector CD8 positive T cells. So in short, when naive T cells encounter professional antigen-presenting cells, such as dendritic cells or macrophages that have antigens processed and presented on their MHC class II molecules, clonal expansion and generation of memory and effector T cells take place. Certain cytokine signals and key transcriptional regulators promote the acquisition of one differentiation state over the other. Memory CD8 positive T cells provide sustainable protective immunity via their ability to generate large waves of effector cells in the face of renewed antigen stimulation. Moreover, they have the ability to rapidly recall effector functions and have broad distribution in peripheral tissues where they can act promptly to participate in tissue immunity. Of note, the population of memory CD8 positive T cells itself is diverse and is often parsed into subsets that vary in functional traits such as cytokine secretion and killing capabilities, longevity, tissue distribution, and migratory behavior. While on the other hand, effector T cells have relatively lower proliferative potential but have higher cytolytic ability compared to memory T cells. So in our research, we were interested in certain subset of memory T cells named as stem-like CD8 positive T cells. In the context of persisting threats, such as chronic infection and progressing cancer, ongoing antigen stimulation of 8 positive T cells largely precludes memory formation and instead drives progressive loss of functionality and eventual deletion in a process called T cell exhaustion or dysfunction. Such chronic CD8 positive T cell responses are replenished by a subset of cells that exhibit some features of classical memory cells generated after acute infection, such as prolonged lifespan, self-renewal capacity, and a level of developmental plasticity. Although previously referred to as memory-like cell populations with such features have since been termed as progenitor exhausted T cells or precursor exhausted T cells. It should be noted that the terms progenitor or precursor have also been used to describe distinct subpopulations of the stem-like cells. So, as mentioned before, stem-like T cells are known for self-renewal, expansion, persistence, and superior anti-tumor responses during chronic inflammation situations. They are characterized with high expression markers or expression of markers such as TCF1, CD44, and PD1 protein, which means that they are activated T cells that have already encountered an antigen. They are generated in draining lymph node, and when they are activated, they differentiate into transitory and terminally differentiated cytotoxic T cells, which then migrate into tumor microenvironment to exert their cytotoxic capabilities. Recent studies have indicated that they are the primary responders of immune checkpoint blockade therapy, such as PD-1 antibody. Thus, emphasis is on the generation and increase of the stem-like T cells in terms of cancer immunotherapeutic efficacy. But the main question is about how we could increase and infiltrate this kind of T cells into the tumor microenvironment in the first place. In hot tumors or immune-inflamed ones, antigen-presenting cells successfully present tumor antigens to T cells in the lymph node. Then these activated T cells easily infiltrate into the tumor microenvironment. But when it comes to cold tumors, activated T cells cannot penetrate the parenchyma of the tumor, or the immunosuppressive nature of the tumor could even inhibit the antigen presentation process, which results in T cell desertification of tumor. So Professor Yong-Sung Go gave us a clue on how to shift the cold tumor microenvironment into a hot one by utilizing gram-negative and gram-positive bacterial extracellular vesicles as a cancer immunotherapeutic agent. So he published a paper in 2017 about extracellular vesicles from gram-negative and gram-positive bacteria that could be the novel platform for cancer immunotherapy. This study showed that bacterial outer membrane vesicles could suppress tumor growth in interferon gamma-mediated anti-tumor response. OMVs showed effective anti-tumor responses in multiple tumor models, such as subcutaneous MC38 tumor model, spontaneous lung metastasis model induced by 4T1 carcinoma and B16BL6 melanoma cell lines, and CT26 mouse colon cancer model. It also showed long-term memory effect, which was confirmed by complete remission of tumor after several re-challenge of CT26 tumor cells upon OMV's treatment. In vivo distribution and targeting ability of OMVs to tumor tissue after systemic administration was observed as well. Moreover, OMVs induced interferon gamma-mediated activation of T cells and NK cells, which was the key mechanism responsible for the effective anti-tumor responses. So we know that OMVs are we know that bacterial OMVs are a very effective agent that could be used to inhibit tumor. However, in terms of developing OMVs as a next-generation cancer immunotherapeutic agent, it is necessary to produce bacterial EVs in a large scale. Thus, in this study, which was recently published in Journal of Extracellular Vesicles, we developed mass production strategies to isolate E. coli OMVs in a large scale and utilized antigen-agnostic strategy where these mass-produced OMVs are used as immunostimulatory agents to modify the tumor microenvironment without the need for antigen prediction. So as a brief summary, we isolated gram-negative bacterial outer membrane vesicles in a large scale, as mentioned before, and perform characterization and proteomic analysis of these mass-produced OMVs. We then examined therapeutic efficacy of E. coli OMVs and examined dose-dependent tumor growth inhibition in various cancer models that has or without severe toxicity. We also characterized tumor-infiltrating lymphocytes, especially T cell subsets that infiltrated into tumor tissue upon OMV's treatment. And moreover, we tested whether E. coli OMVs and anti-PD-1 immunotherapy had synergistic effect when treated together in combination. To start off, I would like to begin with our schematic workflow of large-scale mass production of E. coli OMVs. OMVs were first isolated from 160 liter of conditioned media of E. coli BL21 delta MSBB in a 300 liter scale bioreactor. After pre-clearing the conditioned medium by centrifugation and filtration, OMVs were concentrated by ultrafiltration. OMVs were isolated by metal precipitation and size exclusion chromatography, and then subsequently subjected to filtration and dialysis. So from 160 liter of the conditioned medium, we isolated 357 milligram in total protein amounts of OMVs, which was equivalent to 3.93 times 10 to the 15 particles, or 1.1 times 10 to the 10 particles per microgram in total protein amounts of OMVs. Next, we went on to characterize this mass-produced E. coli OMVs. So transmission electron microscopy allowed us to visualize OMVs that have homogeneously spherical and lipid bilayered structures with average diameter of around 24 nanometers as measured by DLS. And SDS page result demonstrated that OMVs had distinct protein compositions when compared to whole cell lysates. And in addition, outer membrane proteins such as OMP-A were enriched in OMVs, while cytosolic proteins such as FTSZ were de-enriched, as previously reported in other research. Proteomic analysis was performed to further characterize this mass-produced E. coli OMVs. After in-gel digestion of methanol chloroform precipitated OMV proteins with trypsin, the peptides were analyzed by LC-MSMS with 3 technical replicates. The MSMS files were searched with E. coli K12 proteome from UniProt databases using Extendum and transproteomic pipeline. So proteomic analysis revealed a total of 792, 802, and 782 vesicular proteins that were identified from each replicate, resulting in identifying a total of 882 non-redundant vesicular proteins. And their subcellular localizations were determined with UniProt and David databases. The abundance of whole or top 100 vesicular proteins were listed according to their abundance ranks, and we marked different subcellular localizations with different colors in the figure in the middle and on the right one. Among whole vesicular proteins, outer membrane proteins were located at high abundance ranks, and in fact, among top 100 abundant vesicular proteins, there were 32 outer membrane proteins that summed up to 55.2%, followed by cytosolic proteins, inner membrane proteins, and periplasmic proteins. And moreover, we were able to identify some classical OMV marker proteins, such as LAMP-B, OMP-F, LPP, and OMP-A, which ranked top 10 out of a total of 882 vesicular proteins. Next, we went over to examine the therapeutic efficacy of this mass-produced E. coli OMVs, and this is how we designed our animal experiments. So of these 2 schemes, as shown as figure A and B, the first one was done to test the anti-tumor efficacy of E. coli OMVs via measuring tumor growth curve of several tumor models and analyzing various immune cell subsets with flow cytometry analysis. In brief, we first inoculated the tumor cells, and by the time the tumor volume reached about 100 cubic millimeter, we administered E. coli OMVs directly into the tumor in 3 days interval for 4 times. Then flow cytometry analysis was performed 3 days after the final OMV administration. Figure B was done to test the synergistic effect of E. coli OMVs with anti-PD-1 antibody therapy. Everything else was done pretty much the same with the first scheme, except that OMVs was administered 3 times in 3 days interval, while anti-PD-1 antibody was administered together with OMVs for 3 times in 3 days interval, starting from the second OMV administration. So we first tested the anti-tumor efficacy of E. coli OMVs in MB49, which is mouse bladder cancer model in B6 background, and EMT6, which is triple-negative breast cancer model in BALB-C background. For both MB49 and EMT6 tumor model, OMVs induced significant tumor growth inhibition in a dose-dependent manner, where complete remission of both tumor models was observed in the majority of mice treated with the highest dosage, which was 3.52 times 10 to the 9 particles of OMVs. When treated with lower doses, 63% and 94% of tumor growth curve inhibition was achieved in MV49 tumor, whereas 56% and 85% of tumor growth inhibition was achieved in EMT6 tumor. We next evaluated whether E. coli OMVs have any adverse effects on mice by subcutaneously administering different amounts of OMVs into B6 mice. No mice were dead upon administering 1.1 times 10 to the 11 and 3.3 times 10 to the 11 particles of OMVs, which was about 30 times and 100 times higher than the effective dose of OMVs that caused complete remission of 2 tumor models shown in the previous slide. However, a total of 4 mice out of 10 mice were dead upon administering 1.1 times 10 to the 12 particles of OMVs, which is more than 300 times higher than the effective dose of OMVs. The mice experienced temporal loss of body weights in a dose-dependent manner, but they were recovered at most 10 days after OMV administration. In addition, we observed that some mice, especially those administered with the highest dosage of OMVs, showed piloerection, eye exudates, and diarrhea temporally, but the effects disappeared about a week from OMV administration. Next, to evaluate whether OMVs, which do not contain cancer antigens, could directly activate cancer antigen-specific T-cell responses, we inoculated the mice with MC38 ova and B16F10 ova tumor cell lines that were generated to express whole ova protein as a cancer neoantigen. Before evaluating infiltration of cancer-specific T-cell responses, we first performed flow cytometry analysis to look at the general immune profiling of the tumor microenvironment. As a result, we examined that OMV treatment significantly increased infiltration of total CD3 and CD8 positive T cells in MC38 ova tumor model, while increasing infiltration of total CD3, CD4, and CD8 positive T cells in B16F10 ova tumor model. And in both tumor models, OMV treatment decreased the proportion of regulatory T cells, thus significantly increasing the CD8 to Treg ratio. T cells that were infiltrated into the tumor microenvironment upon OMV treatment showed superior effector function compared to that of CD8 positive T cells of vehicle treatment group. To assess cytolytic activity of CD8 positive T cells, we analyzed the frequency of CD8 positive T cells expressing cytotoxin molecules such as granzyme B, TNF alpha, perforin, and interferon gamma. We found out that OMV treatment induced substantial increase in the expression of cytotoxin molecules on CD8 positive T cells in both MC38 ova and B16F10 ova tumor models compared to vehicle treatment. So taken together, we were able to find out that OMV treatment induced increase in infiltration of T cells that has elevated effector function into the tumor tissue. So now that we know more T cells infiltrate into the tumor microenvironment, but we wondered how much of these T cells are specific to the cancer cells. So we next examined whether OMVs could induce activation and recruitment of cancer antigen-specific T cells into the tumor microenvironment using tetramer staining of tumor-associated antigens. So here, REPS1 tetramer was used to stain CD8 positive T cells specific for MC38 tumor cells, while GP100 tetramer was used to analyze B16F10 specific CD8 positive T cells. We found out that cancer-specific CD8 positive T cells were increased in both tumor models by looking at significant increase of REPS1 specific CD8 positive T cells and GP100 specific CD8 positive T cells after OMV treatment. Moreover, OMVs were also capable of increasing the activation and recruitment of T cells that are specific to the neoantigen of the cancer. Here we used Synfeco and Ahania tetramer to analyze OVA-specific CD8 and CD4 positive T cells, and were able to find out that both CD8 and CD4 positive T cells specific to OVA protein were highly increased upon OMV treatment in both tumor models. So OMVs did not only increase the infiltration of tumor-associated antigen-specific CD8 positive T cells, but also increased neoantigen-specific CD8 and CD4 positive T cells into the tumor tissue. To further characterize the phenotype of tumor-infiltrating T cells, we performed flow cytometry analysis to measure the frequency of T cells that exhibit stem-like properties. So evaluation of CD8 positive T cells that express TCF1 marker demonstrated that OMVs significantly increased infiltration of stem-like CD8 positive T cells and neoantigen-specific stem-like CD8 positive T cells in the tumor microenvironment of MC38 OVA tumor model. OMV treatment also increased expression of T cell exhaustion markers, such as PD-1, TIM3, and CD39 molecules on tumor-infiltrating CD8 positive T cells, which indicated that cancer antigen-specific CD8 positive T cells were activated and recruited into the tumor microenvironment and became exhausted due to chronic exposure of tumor antigens. As mentioned briefly in the introduction, exhausted T cells are characterized by the loss of effector functions and sustained upregulation of negative immune checkpoint molecules. And since the interactions between PD-1 and PD-L1 play a major role in regulation of T cell exhaustion, PD-1 and PD-L1 targeted immunotherapy have been widely implicated as potentially effective anti-cancer therapy to reinvigorate and to induce selective proliferation of these exhausted T cells. Thus, high expression of exhaustion markers on T cells upon OMV treatment led us to think about whether OMVs would have any synergistic effect when treated together with anti-PD-1 immunotherapy. So next, we used MC38 OVA tumor model to evaluate the synergistic anti-tumor effect of OMVs and anti-PD-1 combination therapy. Groups of MC38 OVA tumor-bearing mice were either untreated or treated with OMVs or anti-PD-1 antibody alone, or treated in combination of OMVs and anti-PD-1 therapy or antibody. As shown in the tumor growth curve, combination therapy, which is colored in purple, resulted in significant tumor growth inhibition compared to those of groups treated with none or single therapy. Next, flow cytometry analysis demonstrated that combination therapy led to further dramatic increase of tumor-infiltrating CD8 positive T cells and neoantigen-specific CD8 positive T cells that exhibit stem-like memory phenotype. It also enhanced proliferation and cytolytic ability of total CD8 positive T cells and neoantigen-specific CD8 positive T cells as well. And this was evaluated by analyzing expression level of Ki67 and cytotoxin molecules such as coenzyme B, perforin, and interferon gamma. Lastly, immunofluorescence microscopy confirmed that OMVs and anti-PD-1 combination therapy increased CD8 positive T cell infiltration while decreased tumor-infiltrating CD11B positive myeloid cell population compared to OMV-only treatment group. So taken together, these results demonstrate that anti-PD-1 therapy synergistically enhances anti-tumor effects induced by OMVs. So to conclude, our research was about development of a new strategy for mass production of E. coli OMVs for us to improve OMVs as a next generation agent for cancer immunotherapy. We were able to isolate total of 357 milligram of OMVs that showed similar morphology and diameter with other bacterial extracellular vesicles. We found no severe toxicity when mice were treated with OMVs dosage that were more than 100 times higher than the effective dose. And this antigen-agnostic OMV treatment induced activation and recruitment of cancer antigen-specific stem-like T cells, which were even enhanced with proliferation and cytolytic ability when treated in combination with anti-PD-1 immunotherapy. And that's it for me. I'd like to thank everyone who participated in this study, especially my professor, Dong-Seop Lee, and our lab members who helped me a lot with all the animal experiments. And of course, I'd like to thank Professor Go and his lab members in Pohang University of Science and Technology and SL Baijen for doing all the works related to OMV's mass production, characterization, and proteomic analysis. Thank you for listening, and I'm now open to any questions.
Thanks so much, Sol, for this really wonderful presentation. It was quite clear and concise, and I think conveyed all of the great work that you've done here. And you've shown some really remarkable achievements with reducing that tumor volume with the OMVs themselves, and especially in the combination. So very impressive, and congratulations on that. I would like to invite our participants now to join with discussion. I have a few questions myself as well. So if anybody has some questions, just please put them in the chat box, or you can also just raise your hand and I can call on you. But I would like to start with a few questions that have come to mind for me. So one of my questions has to do with the source of the bacterial EMVs that you've chosen, which is the E. coli. Is this going to be something that you think will happen for all OMVs from the gram-negative bacterial species, or do you think that there's anything that would be specific to a particular species of bacterium?
That's a very wonderful question. The answer, I'm not really sure, but based on what I know, I believe the mechanism by how our E. coli OMVs worked was there was something to do with the endotoxins in the outer membrane vesicles, or the surface of the outer membrane vesicles. So I don't know if it's going to be same for all strains of other gram-negative bacteria, but I think there should be an effect related to the endotoxins of LPS with all the gram-negative bacteria, because they could activate the innate immune responses by pathways such as TLR pathways. And yes, I think not to full extent, but they still have, or there might be effect that correlates or all strains of E. coli OMVs.
Fantastic. Yeah. So it's that stimulation of the innate immune responses that might be the key here. Yeah. Good. Well, I see. Let's see. Hava Erzgin Kilgers, you have a question. Could you, if you'd like to ask that yourself, please go ahead. So maybe that Hava does not have the microphone. So thank you for this great presentation. I was wondering how you administer the anti-PD-1 therapy into the mice. Was there any encapsulation? What was the formulation there, Sol?
Oh, so there wasn't any encapsulations. We just treated the anti-PD-1 antibody itself via intravenous route. Yeah, and that was it.
So the anti-PD-1 antibody that went right into the circulation, and whereas you were injecting the OMVs into the tumor itself, correct?
Yes, that's right.
Yes. Okay. Great. Okay. So hopefully that answers your question, Hava. And I see that your microphone is not working, so please feel free to follow up if you have additional comments or questions. Now, you mentioned that you think that this is probably the endotoxin that is producing the effect. Would it be just as effective to just put the endotoxin itself into the tumor, or do you feel that the OMVs are really presenting a kind of signaling platform that might be enhancing the delivery, the presentation of that endotoxin?
That's an interesting question. So, so
And
I
maybe I'll
think,
just, if you think about the background of what we know about how EVs are presenting things, they can present a molecule, an antigen, a molecular pattern, basically in a context, in a specific context. And so, sometimes we think about the EV as sort of a combinatorial presentation platform. And
mhm.
I'm just wondering if you think that that's the case for the bacterial OMVs, not just mammalian EVs.
um, I think, I think the most possible reason or for this whole activation of innate immune responses or adaptive immune responses was mainly due to the endotoxins or the LPS. I'm pretty sure there are other pathways that could interact with the with the cells of innate immune responses. But based on what I know, I think the main pathway or the main reason for this activation or the most important or the most pathway that contributes the most would be the endotoxins. I, I hope that answered your question.
Yeah, no, that's great. Thank you. Let's see here. I did have a question that came in to me through the chat box privately, but so I'll ask that myself as well. So the question here is, what if you were to give more injections? So I guess if I recall correctly, you were giving 3 injections into the tumors of the bacterial OMVs. Uh,
Mm.
and in a cancer patient, it might be necessary to have an even more extended treatment course. So I guess the question is, do you think that more injections would enhance the effect, or would you see diminishing returns with increasing injections?
Um, for us, since we only do mouse or mouse, we um, I think that's something to do with the toxicity of OMVs when it goes to the patients. But based on what we observed, toxicity was diminished after a few injections of OMVs. So I think toxicity would be fine. But based on in terms of immune responses, I think more injections of OMVs would increase or sustain the activation of adaptive immune responses. But we were not able to find out whether more injections of OMVs could increase the adaptive immune response because by the time we injected like 3 to 4 times of OMVs with high dosage, the tumors were completely um, inhibited. So yes.
And now that you have finished and published these findings, what do you see as next steps that are going to take you towards human patient therapies?
Human patient therapies. I think the first thing would be with the toxicity. Even though the endotoxins are attenuated in our bacterial OMVs, we cannot say, or as shown in the previous slides, there are still remaining toxicities that might be dangerous or that might be sublethal or when it comes to patients. Since we only do in animals, these kind of this extent of toxicity could be fine or okay. But our next step should be about reducing the toxicity, I guess, and to yes, to make our OMVs even safer.
Yeah, indeed. So So in my group, we've used some bacterial OMVs as a vaccine platform, and you're probably very well familiar with work that has also been done in Korea towards this end, um, for stimulating the innate immune, or sorry, the adaptive immune system as well. So is it possible, do you think, that we could use similar strategies of putting specific molecules in the surface of the bacterial OMVs, not necessarily as a vaccine, but rather to stimulate particular pathways in those CD8 positive T cells? In other words, would it be possible to make engineered OMVs that might have an even greater effect on the immune cells?
I think 100% possible. And I think that's the whole bacterial OMVs. They, since they have around the surface of, like, since the endotoxins itself could stimulate the innate immune responses, could engineer the OMVs to activate other pathways, such as adaptive immune responses, or to target T cells that could even more sustain the activation of T cells, or to kind of make engineer the OMVs to make T cells differentiate into other subset of T cells that's even more powerful. Yes, I think that's 100% possible, and that's a great question. Thank you.
Great. Yeah. So it looks like there's a lot of promise here. There's also a lot to investigate to make these sorts of therapies even more effective. And so I don't see any more questions in the chat box. So we can probably conclude for this session of the EV Club. But Solchan, I wanted to thank you so much for presenting to us today. And of course, most of all, for this exciting work. And also many thanks to the advisors here, to Dong-Sup Lee and Taeyong Lee, and also to Yongsung Go, for making it possible to have this exciting science, but also to share on the EV Club. So so thanks again, Sol. Thanks to everybody who's joined here, and we look forward to also sharing this on the YouTube channel, where I'm sure that there will be a lot of interest in this highly effective therapy. So thanks again, everybody, and hope you have a great rest of your week. And we look forward to seeing you again on a future EV Club. Thanks.
Thank you.